Dear colleagues, my name is Ekaterina Mironova. I am PhD and head of Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Pulmonology, and also Senior Researcher of Laboratory of Molecular Mechanism of Aging, St. Petersburg Institute of Bioregulation and Gerontology. Today I will tell you about peptides affect stem cells aging. Aging is considered to be a multifactorial mechanism which is accompanied by a progressive decrease in the pool of endogenous internal stem cells, as well as damage to their structure and functions, with age increasing. Decreased stem cells regeneration contributed to the development of age-related diseases. Decreased regenerative capacity of endogenous stem cells is regulated due to reduced energy metabolism, disruption of DNA repair, slowing down of RNA and protein synthesis, and accumulation of cellular damage. All cells in the body are formed from stem cells. That's why stem cells function is important for understanding the aging process at the tissue and body levels. Senescent cells affect neighboring cells by releasing certain signaling molecules, which triggers a cascade of intercellular signaling events. It is believed that cellular aging is one of the mechanisms of the whole organism aging. It is important to note that the loss of proliferative activity due to stem cells aging is equivalent to the loss of their ability to regenerate damaged tissues. This highlights the need to study the mechanism of stem cell aging. Short peptides have been shown to regulate function, activity, proliferation, apoptosis, and stem cells differentiation. Based on the amino acid composition analysis of the endolycan polypeptide complex isolated from the pineal gland of young animals, and the tetrapeptide epitolone was synthesized. You can see it on this graph. From the Wentworth polypeptide complex, which was isolated from the vessels of young animals, the tripeptide vasogen was synthesized. You also can see it on this picture. Epitolone and vasogen protected stem cells from replicative senescence by reducing genes expressions and P16P21 protein synthesis, which is responsible for the cell cycle. Replicative senescence is a process of cell aging with an increase of cell culture passages number. Passage is the transfer of cells from one culture vessel to another for reproduction and accumulation of the cell mass required amount. Based on the amino acid composition analysis of the sycomere polypeptide complex isolated from cartilage and bone tissues of young animals, the tripeptide cartilax was synthesized. It was found that cartilax increased the functional activity of skin fibroblast stem cells during aging by increasing gene expression and dermal fibroblast differentiation proteins in grade 1 PDGFR alpha, SPRI4 twist2 synthesis. Two models of stem cells aging were studied: replicative aging, aging of cells by passages, and stationary aging caused by contact inhibition of cells within one passage. It was found that vasogen contributed to changes in the expression of EGF1, FOXO1, TERT, TNKS2, and NF-kappa B genes, responsible for increasing cell lifespan in two models of cellular aging. Cartilax contributed to the change in the expression of EGF1 and NF-kappa B genes in two models of cellular aging, as well as the third TNFKS2 genes during stationary aging. The KED peptide contributed to changes in the expression of the EGF1 ENF-kappa B genes in 2 aging models, as well as the TERT and FOXA1 genes during stationary aging. Long-term cultivation of stem cells is necessary for clinical use in order to regenerate tissues and organs in cell replacement therapy. Thus, short peptides are potential substances that can be used as additional agents for maintaining the stem cell's morphology and functions, as well as reducing the aging and apoptosis markers expression. Thank you for your attention.